So, we will talk a little bit about, but we have mentioned already, choosing the control intervention, how we do randomization, protecting the random allocation, and then this aspect of study integrity and making sure that the study is done as per the protocol is being done. And this is where the complexity comes in. So the design is important, but ensuring the study integrity when you're dealing with humans is not an easy task to do. So what is the optimal control intervention? Where do you get this intervention that you're going to do? You have dreamt this. You have thought, oh, I think this intervention that we do in our clinic is actually really good. Maybe this is the way we should do the open heart surgery. We have to do this. We had one physician that decided, once I have somebody open, there's this left atrium appendage that is pointless there. And maybe I think it's causing atrial fibrillation if I don't remove it. It's like removing the appendix. Why don't I just remove this thing? No one would fund it. But he got a pilot money to do it, and it showed it worked, and it actually is beneficial. And so that changed practice on it. So where do you come up with these ideas of what is the optimal intervention to do? We already showed that we have these other options, and usually we cannot do new intervention versus placebo. We do it versus usual care. But now we also think about best available care, because usual care in one place may not be best available care in another place. And so we have to worry about this. Who worries about it? You, when you're designing your study. Your ethics committee will be saying, you're doing usual care, but usual care that's done in Europe, usual care that's done in India, or usual care that's done in China, or whatever. What is usual care? Does every place in India do the same usual care? Usual care in CMC may not be the same as usual care in some rural area, for example. So then, what is it that you do? So you have to decide on these things. Most people will do new intervention plus usual care versus usual care. So now you're seeing what is the additional contribution of your new intervention to usual care. Or they will say that we're going to do it in a non-inferiority one if usual care is something that works, which we already talked about as well. So there's a nice paper that came out in 2007 on should you use placebo, should you use best available therapy in the control arm in clinical trials. And that's part of what you need to worry about when you're designing your study. Now, the main word in the clinical trial is randomized. And it's actually not randomized clinical trial, it's randomized controlled trial. Okay? So that's the control group. They don't have to be only in clinical applications. It's a control design. But we randomize. So options for non-random allocation should never do historical controls. That's garbage. You should not assign people yourself because you have personal bias as to who gets what. Systematic assignment, is it wrong or is it right? You could say, well, people have an ID number, and those that end in an odd number will go to this, those that end in an even number will go that. That's objective. That's random. Whoever gets your number is random. It may be random, it may be objective, but it has a problem. It's discoverable. You know that, oh, this person has an even one, and this person is going to go to A. But this person has an odd one, so this person is going to B. And if I know what you're going to be getting, I can use my bias because I say, oh, he's a little bit more frail. He meets the addition, but I don't want him to go into the intervention that I think is going to work. So I said, oh, sorry, I don't even mention to him that we're doing a trial. But he doesn't go in. You can manipulate things if it's discovered. So we don't ever do systematic assignment anymore. People used to do it. So what is achieved by randomization? I think you've already said it. Everybody has an equal or known probability of being offered the intervention, and all potential confounders, known and unknown, will be equally distributed in the long run or in large sample sizes. And so this is the argument that we have. Statistical test assumptions are met and things work. Now, usually we do equal random allocation. So 50-50 chance to go to A or to B. Some people say, like in those villages, they had 5 villages that went to intervention, 10 that went to control. Why did they do unequal? I don't know. They didn't say in the paper. I think it is because of cost and effort that they did it. But why would we do an equal allocation? Maybe because of practical reasons. But if you think that one is better than the other, and therefore you want to give the one that's better to more people, or you think that's better to more people, that's when you would do it. So then we come back to, do you have equipoise when you're doing that? If you think that I should give one is better already. People, they say, well, it's not what you think, it's what the people think. So if the people you're randomizing think that the new one is better, even it may not be, in order for you to recruit these people, you may want to say, you know, you have 2/3 chance of going to this one, and you have only 1/3 chance of staying in the usual care. So you have a better chance of getting the new intervention, which may be better. We don't know, but you have it. It helps in recruitment, but usually is used as a way to help in recruitment. So you have then maybe more people are going to intervention on that. And so this sometimes we do unbalanced randomization. And randomization is operationalized by, it used to be in the old days, by throwing dice or putting numbers in a roulette, a table of random numbers, or nowadays we use computer programs that give us a pseudo-random number, but some programs are better than others to give us a pseudo-random numbers, and we generate these lists based on that. Now, you may have heard of blocking, and blocking is an interesting technique because suppose I'm going, let's take a small example. If I'm going to randomize 20 people, 10 and 10, and I'm going to do it by simple randomization. So I have 20 people that I'm going to randomize. So from 0, 1 up to 20. And I'm going to toss a coin. This goes to A, and the next one goes to A, and the next one goes to A. Can it happen? Of course. Keep going like that, and sometimes maybe this one goes to B, then goes to A, for some reason, then B, B, A, B, A. 14, 15, 16, 17. How many A's? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11A, 9B. It can happen. Well, say, well, people will not think I know what I'm doing if I do that. So why don't I do block? I do a block of 20. That means that once I got 10A, I got 10A here, the rest of them are B. So I get 10 and 10. That's a block of 20. But what happens here? Bad luck. Suppose my intervention requires that the staff perform some procedures like that study that was done in managed third stage of labor. So they've learned the technique of adequate management of the third stage of labor, which they did not do before. So yeah, they've been trained, they've been certified, they said they're fine, we said they're fine, but it's human nature. The more you do something, the better you get, right? You learn by experience to do things properly. You may follow the protocol, you do all of this stuff, but the more you do it, the better you are. So you're not well trained yet, and the first three people suffered with A. And the last 5 people got B. Oh, so maybe my training now and my experience is not equal for A and B. So what should I do? Well, I can block. I can say, after every 4, I want to have enough 2 A's and 2 B's, for example. Not wait for a block of 20, but do it early on. So what possible blocks do I have? I can have A, B, B, A, B, A, B, A, B, B, A, B, A, B, B, A, B, A, B, B, A. I have 6 possible ones. I'm going to have 12 people, right? I mean, sorry, 20 people in blocks of 4. And I'll give this a number 1, 2, 3, 4, 5, 6. And instead of tossing a coin every time, I'm going to toss a dice. And how many times? So I have 6 possibilities. I'm sorry, I have 4 blocks of 4, and I have 20, so I'm going to need 5 blocks. So I toss the dice 5 times. And I toss it the first time, I get 5. Second time, I get 6. I toss it. Next time, I get 1. Next time I toss it, I get 1 again. And next time I toss it, and I get 6. So now my randomization is not going to be like this. It's going to be, I take 5, I take B, A, B, A, then I take 6, so it's B, B, A, A, then I take 1, A, B, B, 1 again, A, B, B, and then this one, B, B, A. I have 10 and 10, and after every few, there's balance between A's and B's, so the experiences are okay. And this is fine. This is one way to do it. But the statistician will develop this, and we have to hide it from you. Why? Because you are human beings, and you don't like secrets. And you would like to know, oh, is the next person going to go to A or B? Why? Because you have your theory that B is better than A, and I want to show superiority of B to A. And so if you discover that the next one is going to be A or B, you can do the same thing that you could do in the systematic. You can manipulate who goes or not. Because you say, okay, the next person is going to be eligible, and oh, but this fellow is coming tomorrow and he's eligible, but I don't want him there because he may spoil my study. So then you don't even call him tomorrow. Or you say, oh, please come next week. Why? Because if you don't know, but A and B may cause, this is what's there. A and B may cause some side effect. A may give you a little bit more nausea than B. You, as a physician, you know that because you know what you're doing as an intervention. You also know you are treating this person. The person comes to you and says, oh, I had some nausea. He's probably getting B. Another one don't complain, he's probably getting A. Why? You don't want to have secrets. You're told that you are supposed to be blinded to what people are getting, but you are humans also. You don't like to be told secrets. You will try to figure it out, and you're smart enough to figure it out. and you will try to guess. So you know, oh, wait a second. These two did not come. This one complained. These two people, there's two people that have complained. Oh, the next one is going to be A. If you know the block size, and you can cleverly figure out 3 have gone by, this one is A. So now we don't even tell you the block size. And we change the block size. So we use blocks of size 2, blocks of size 4, blocks of size 6. And blocks of size 2, there is AB, BA. Blocks of size 6, AB, there's actually 20 of those. I'm not going to write them out. And now you cannot guess what you're getting. Because you may get, for example, the first block, now we have to choose. We randomly choose, toss a 3-sided coin, and say number 2 came first. Then I toss another one. Oh, number 4 came later. And then another 2, then a 6 came, and then a 4, and then a 6, something like that. And now I have 2, and I toss a coin, and I get AB. So now I have AB. And no, next one is 4, and I toss a die, and I get number 6. So I get B, B, A, and then I do this, and I get something and keep on. Now, it's hard for you to guess what is coming. And we do this, and we call it permuted blocks. And all you investigators need to know is that we did permuted block randomization. Don't ask questions. What block size did you use? We won't tell you. There is absolutely no reason for you to know except to try to cheat. So therefore, we keep you from doing it. Okay? That's what permuted blocks is. Nothing hard. Now, this is when we do a fixed randomization schedule. So we develop the randomization schedule, and we have these 20, and whatever it turns out to be, we take it. And in the old days, we would hide it in what? We would take 20 envelopes that were opaque envelopes, and you could not check with the light. And we would put in envelope 01, envelope 02, 3, etc. And the next patient comes, and you pick up this, and you open it, and you see them. And that was called allocation concealment. We conceal the allocation until the moment of randomization. So you don't discover it before. That's the only reason. Nowadays, most places, we don't do it with little envelopes unless it's a remote rural area that has no Wi-Fi, no cell phone coverage, no nothing. Because then they need to have something there. Nowadays, we do it on the computer. And what you do is on your mobile, you will have access. Oh, this person is eligible. You put in their screening ID. You put in all the questions for eligibility, all the questions for exclusion. This person is okay. It meets all the criteria. Doesn't need to be excluded. Has this person consented to participate? Have they signed the consent form? They say yes. And once there says, if the person says yes, this person will be automatically randomized. And then you say yes, and then you get the answer. The envelope is opened by the computer, and it says B. That's it. Or actually, it doesn't even say B. It may say, give them packet number 42. That's it. Because packet number 42, if somebody looks at it and says, oh, this looks like, if it says B, and you know that B, it looks like it's the pill, it's not exactly the same as the other one. It has slightly different something, then people will just unblind the whole study. So we never really do that. We just say, give them packet 42, this one give them packet 49, give them packet this, give them packet that, and that's how it works, okay? So we try to avoid these bias in it. And this is what blocking achieves the balance. Now, blocking is just to achieve balance. We don't care to analyze by block. So it's not a stratum. Okay? And so here are the blocks, for example, of size 4 in equal size ones. And then we do the blocks of size 2, 4, and 6, as I said, and these are the ones listed. So it makes it harder for you to guess what is the next one. Everybody okay on permuted blocks? So in permuted blocks is a two-step process to create the randomization schedule. We first randomly select a block size. And here, for example, I use blocks of size 2, 4, and 6. We may use other sizes. We may use 4 and 8. If it's a 3-arm study, we'll use 3, 6, 9, for example. And we first randomly select which block size in a random order. And then we randomly select which order set within the block. So, for example, in this one, we had 2, 4, 2, 6. So maybe 2, I may get the AB, and then with the 4, I may get the BBAA. Then with the 2, I get the AB, and then with the 6, I may get, who knows, B, 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 A, and so forth like that. So you constructed that. Okay. Another way of randomizing is when you do this, all you're doing is balancing the number of people that get A and the number of people that get B. But what if the first people you get are female, 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 male, male, etc. You may end up with an imbalance in sex between the two of them. If you're doing large numbers, that's not going to happen. If you're doing a study with 20,000, this will be balanced. But when you're doing studies of a few hundred, there may be some things that you think are important confounders that you want to make sure they're balanced. And so one way to do that is to create strata. And let's say the strata you want to have are, I want to have, in my study, people that present with this condition, there's 10% that present with very severe symptoms. About 70% present with okay, moderate to severe, and about 20% present with mild symptoms. So I'm going to make a stratum of the severe, the moderate, and the mild. And it's at first come, oh, this person is severe. Then I randomize within the severe. I randomize within the mild, the moderate, and within the mild. So then here, the distribution of severity will be comparable to the distribution of severity here. And so the purpose of randomization that is stratified is to assure that I get balance on important confounders that I want to have, especially when I do a small study. And small may be around 200. Large is over 10,000, 20,000, etc. But most people cannot do those huge studies. You do studies of about maybe 2,000, 3,000 sometimes, but most people do 400, 500. So not everything is going to be balanced. So you identify beforehand what is a very important factor for my disease that I'm studying, that I really want to have that one. So you want to make sure that it's balanced. Now, there's two purposes of stratification. One is this one. I want to get balance, appropriate balance in the two arms. But the other one may be, oh, wait a second. I want to know, is my intervention more effective in the severe or more effective in this group or that group? Like the males or in the females or in the young people, the old people, in the more severe, the less severe. If you're interested in having an analysis within a stratum, now you have to have sufficient sample size within that stratum. So it's not, so if the purpose of stratification is just to get balance, you don't have to worry about it. But if you're interested in saying at the end of the day, oh, my treatment is such and such in this group and that group, you better have enough numbers in it. Otherwise, you will not get a conclusive result. So when to stratify? When you had one or two or few baseline characteristics that are strongly related to the outcome, so potential confounders, and you have small trials, or especially in cluster randomized trials, because we are not randomizing a lot of people. We have then, for a lot of villages, or a lot of schools, you want to stratify on the important things. And realize that when we do large studies, like we were going to have to do multi-center studies, we randomize within center. And within center randomization is essentially stratifying by center. Stratified randomization by center. We don't care to give you a result of my intervention in each center, so I don't have to have adequate numbers in each center, but any center effect is going to be balanced between A and B. That's why we do the randomization within center in large multi-center trials. Now, special case of randomization that is stratified is matching one, where you now match one-on-one. For example, in twin studies, we have these matched pairs. And so each one within a twin set, one of them goes to intervention, one of them goes to control, for example. And that's a very restricted randomization. And you may be interested in, the controlling for genetic variability by doing this, for example. And this is one way to, now the distribution of this genetic issue is comparable between the intervention and the control. So these pair studies, or match pair studies of twins were fairly common if you're studying something that may be of interest with genetics. And then we talked about clustered randomization already, so I won't talk about that again. Now, with cluster randomization, what happens, or when you have few units being randomized, especially in cluster randomization, you don't get the benefit of randomizing a lot of units. We have few clusters, and so things are not going to be balanced. And if things are not going to be balanced, what do I do? Oh, I have maybe 16, or we had, 24 hospitals in Uruguay and Argentina. They had 4 in Uruguay and 20 in Argentina. So they said, okay, we better randomize within country. So we want 2 going to intervention in Uruguay and 2 to control, and 10 in Argentina going to intervention and 10 to control. So that takes care of country as a potential confounder. So what about we do 1, hospital size, obstetric team experience, the size of the obstetric team, the volume of deliveries they have. They had like 20, 30 different variables that they thought that would affect the outcome of the intervention. We can't stratify all of these things. And we may not get balance because there may be 3 of those hospitals in Uruguay are big ones and 1 is small. So then what do? I cannot get 2 and 2, 2 big and 2 small in each one, because I only have 1 small. So you cannot do these things. So you cannot do a lot of the stratification to balance things when you have multiple things to stratify. You can maybe do it on 1 thing or 2 things. So what has been proposed is restricted randomization. And restricted randomization, one of those is minimization. And this is a very clever design that this professor did way back 1974. It was almost 50 years ago he proposed this. And what he said is following. Suppose I have these 20, well, no, let's do less than 20. That's too many. Let's say I have 8. I have 8 clinics or 8 clusters. Okay, so I want to have 4 to intervention and 4 to control. Okay, how many possible ways are there? And I want to balance them on x1, x2, x3, x4, x5. These are known confounders. And I have all of these. Wow, there's too many. We can't really do it very well, but let's try. What he suggested is, if I have 8 clusters and I'm going to do 4 to A and 4 to B, I have 8 choose 4 possible combinations. And so this is 8 factorial divided by 4 factorial, 4 factorial. So that's 8 times 7 times 6 times 5 times 4 times 3 times 2 times 1, because the other will cancel out with the other. So that's 8 cancels. This is 2. So I have 35. I have 70. I have 70 possible ways to do it. So I can do 70 different ways of giving, let's say, whatever, let's call them. Here I have A, B, C, D, E, F, G, H are the hospitals. And so I could do that. And then I could do A, B, C, E, and put D on this side, F, G, H. And I can do all of them. And I can have eventually E, F, G, H to A, B, C, D. Do I have, well, yeah. And so if I do this, now I can see, oh, wait a second. What is the distribution of x1, x2, x3, x4, x5 in this group? And what is it in this group? And he created a linear discriminant function that says, how similar are these to these? How similar are these to these? And he had a similarity in this linear discriminant function. Let the computer generate it. So now I get the similarity for all of this. And this is similarity 1, 0, 1, similarity 0, 2, similarity 70. And I have all the similarities. Well, the one that is the most similar has the least difference between them. So take the smallest difference between them. That's the most similar. And let's say, randomization sequence number 37, whatever it turns out to be, this one was the best. The one that minimized the differences between them. That's the best one. Choose it. Let's do that. Fantastic. That's what minimization is. Nobody uses it. Why does nobody use it? Because the word randomization is not there. They chose one. No matter what criteria you did not randomize. What do we do? So somebody else clever also said, you know what we should do? We calculate the 70 similarities and let's put a threshold of tolerability and say, if that similarity is less than some value, then it's okay. Similar or greater than some value. The difference is less than some. So maybe out of those, 0.4 was okay, 0.7 was okay, 13 was okay, 22 was okay, 37 was okay, 39 was okay, 42 was okay, and 70 was okay. Now I have 1, 2, 3, 4, 5, 6, 7, 8. 8 out of the 70 I will tolerate. These are sufficiently similar that any one of those is okay with me. Now I flip a coin and see which one of those 8 works. So we call this restricted randomization. The word randomization is there. So, or people call it optimized randomization or covariate constrained randomization. And this is actually what these people in Argentina and Uruguay did. They describe it in their paper in BMC Women's Health, but they didn't have 8. They had 20 in one place and 4 in the other. And so they had 24. So if you take 24 factorial, 12 factorial, 12 factorial, that is an enormous bazillion, gazillion, trillion numbers. And they put a criterion on it that gave them about something like 37 billion would be okay. And they randomly selected 1 out of those 37 billion. The computer does this stuff. There's absolutely no problem with the computer doing this stuff. But people do this. And so it is a way to do optimized randomization. And if you've heard the word covariate constrained randomization, these are the covariates, x1, x2, x3, x4, x5. The only benefit of this is that you're able to say it's randomized, which is fine. But again, you've only controlled, randomized on things that you know are potential confounders. You don't know, you didn't balance everything because there are unknown things that you may have not been able to, but that's just a limitation of this thing. Now, the more attractive one is this adaptive randomization design. Here comes the word adaptive again, because what everybody loves adaptive, because I can change things and do things and adapt things. I don't have to be ruled by something. So this is now when results of previous subjects is used to adjust the allocation of subsequent patients. Now, this is usually done almost never in large-scale trials, almost never in phase 3 trials, never in chronic diseases, because you've got to wait to get the outcome of one patient before you know, oh, now I have to do the next patient. So this is impossible to do in big studies. The advantage is this is used in phase 1 trials, which are not really randomized control ones. And sometimes in those finding phase 2 trials. So what they do, for example, is they call this play the winner. And so you start off and you flip a coin and the first person gets A. And if they get a positive result on A, the next person gets A automatically. But if this person gets a negative result on A, the next person gets B. And if this person that got B gets a negative result, then the next one is shift to A to the next person. If this gets positive, the next one gets A. If this one is positive, the next one gets A. And whenever this gets negative, it goes to B. And it goes towards the one that is winning. This way, more people get the one that is working better. B is never working as good, more people get A. And it's called play the winner. They also do things like bias coin. Bias coin is you start off with an unbiased coin, half and half, 50-50. You toss it, and each person has equal probability of going to A or B. So let's say they go to A, but now there's one person in A, so you increase the probability for the second person to go to B. So now this is 1/3, 2/3. And let's say you flip this bias coin, or the computer does it, and it says, oh, you go to A. So now you have 2 people in A, and you have nobody in B. So you um, um, increase the probability of getting B in the next term. You bias it even more. So now it's 3/4 to 1/4, and you flip this bias coin, and it comes out B. It goes B. So now you have 1 in B, 2 in A. You still want to get more probability for B than A, but now you increment A to 2/5, and this is 3/5. And now you flip this coin, and you get B. So now you have 2B and 2A. So the next one is equal, 3/6. And now you get B. What happens in the next one? What should it be here? If you got B here, what would have been here? This would have been 4/7 to 3/7, right? And then I got B, so then what would happen there? I would have even more for A, right? So it'd be 5/8 to 3/8. And they keep doing this. But it's only possible, as I said, when you have quick results and you get them quickly. And you are doing this usually in the phase 2 trials in a laboratory, in a pharma company, where they get the results quickly and they are unblinded. They know which one is winning as it's going along. So these are never done really in those big trials that we've been talking about, the phase 3 trials. It's only done in phase 2 trials. Okay. So minimization is you calculate all possible allocations. So if you have n centers, then it's n factorial centers that half of them are going to go to A and half of them are going to go to B. So you have half of them factorial and half of them factorial, give you all possible combinations. This is just combinatorics theory to get there. You don't calculate it, statistician will calculate it for you. But then once you have those all possibilities, you have to create some function that is meaningful to you. The statistician will help you build it, that says these groups are similar, these ones here are similar to these ones here, or they're very different. And you have a different discrepancy. Yeah, I call it a discrepancy index. There's one minus a similarity index, but it's a discrepancy index. And you minimize the discrepancy. And the allocation that gives you the smallest one out of all the possibilities is the minimal allocation that works the best, because it makes the two groups, the A group and the B group, similar. The most similar is the least discrepant. Okay? And so that's the minimization. And the restricted or covariate constraint, sorry, is where you don't just select the smallest one, you select a group of small ones that you will tolerate. So you set the boundary. You say, okay, I'm, as an investigator, I'm willing to tolerate this much discrepancy, but not larger than this. And now at that point, you do this and select that subset where the allocations are tolerable below your threshold, and then you randomly select one. That's the only difference between them. But the reason this one is used is because it has the word randomization. Or that's the reason the other one is not used, because it doesn't have the word randomization. And people will not like anything that is not randomized. Does that help?
The allocation is equal.
No.
No.
This one has a
every,
different,
every patient, every person has an equal chance of getting allocated to
well,
treatment.
not here, not here. Here it's a completely different mindset. Here is, you are now testing. I want to see if A is better than B in not a big controlled trial. This is in a small study done in the pharma company to see whether there is some evidence that A is better than B. You don't get these things published. usually, because pharma doesn't want to tell you secrets about which things did not work well. And a lot of things did not work. But this is one way to get volunteers to come to their study, because they'll say, you have a good chance of getting the best one because we will be giving you the best one. That's why we use it. Very good. It helps recruitment. So they will end to recruitment is a good question. Once there are different reasons for it, right? So they will just say how many people eventually got positive in A versus positive in B. And if there are ways that you would set up that will say, if I get, you can calculate the proportion of success in A and proportion of success in B. In just the same way, they will say, if we have this much more success, then we'll stop, or we'll continue it as long as that. Here I have very dramatic, nothing works with B. So you should probably stop very early.
Experiment, the biased point design, the weightage of the coin is based on the assignment of the result, because if it's based on the assignment, then it's the same as blocking is what I thought. Like, we're just making sure that it's equal numbers of A's and B's.
Yeah, so that's a good question. They do it both ways. This one is based on balancing things. And so it's not exactly the same as blocking, but yes, it is trying to get enough numbers balanced between them. And it's an alternative to blocking that also keeps people from guessing what is going to be happening. But some people will do it based on the outcome. If you do it based on the outcome, it's the play in the winner. All right. So we try to randomize. And again, the object of randomization is to ensure that you have balance on confounders. Balance in numbers is one, but balance in confounders. But maybe things work, maybe things do not work. And so what you try to do is check to see if balance is achieved. And how do you do it? Famous table 1 of every clinical trial is baseline comparison. Okay. And if you have small sample sizes, I can almost guarantee that there'll be some things that will not be balanced. That's your bad luck. And you may have to decide whether you want to adjust the analysis, the secondary analysis, based on this imbalances. You're usually not allowed to adjust by covariates in your primary analysis. But it could be that masking or blinding, the reason we say masking nowadays is because when I worked with ophthalmologists and I said, oh, is this a double-blind study? We don't say that word here. That's a bad word. So we use masking. All right, fine. We'll use masking. If people can discover and patients get to know because of side effects or because of how they're feeling, they guess that they are this, that can change their behavior. If physicians or clinicians that are monitoring the patient get to guess, and they have a better chance of guessing because they know more about the disease, the side effects, etc., the behavior, the outcomes. They guess, they can change the behavior too. Or if somebody actually cheated and was able to get those envelopes and see things and change things or do whatever, then things can go wrong. So we check it by reviewing table 1 of variables known and measured. But I can guarantee you that if you do less than 100 subjects, simple randomization may not always work. And that if you do more than 10,000, simple randomization almost always will work. But as a statistician, I never say certain. I don't say simple randomization works. Almost always works. There's always a case where it may not, by pure bad luck. And here, it may not work, but it may work. Also, you may be lucky, or you may be unlucky. but you want to improve your chances of being lucky by doing more sample size. So, I'm going backwards. Okay, so that was randomization. Now we're doing the study. We're going to be, we have hopefully designed a study that is well designed. In fact, we can think about a study that is poorly designed as an unethical thing to do. Because if you design your study poorly, then the result you're going to get is going to be what? You're going to say either this is superior when it's not, or this is not superior when it is superior. And you will then, if it's an important study, and hopefully you're studying something that is important, not a waste of time, you're going to potentially change practice when you shouldn't, or you're potentially going to not change practices when you should. So it's unethical to design a study poorly. And the ethics committees many times think about it that way. Is this a study that is ethically is designed so that it will be ethical at the end? But then you may design your study well, but if you don't conduct your study well, that's also unethical, because you will get sloppiness, will lead to saying maybe there is no meaningful difference between them, but it's not because the effect was not there. It's because you have so much noise, you have so much missing data, you had so much garbage there because you did the study wrong. You didn't monitor things, your staff did not comply, the patients did not adhere, people did not come in, nothing was done well. And all of these things are important. So one is maintaining objectivity, and the other one is maintaining high quality of all the processes involved there. The training, the certification of the staff, supervision of the staff, checking, monitoring them, avoid possibility of tampering documentation, and the privacy of the data. So preserving objectivity in randomization is done by concealing the allocation before you do the random allocation, and then you maintain it with the masking of the investigator. So people do not include their personal bias into this. So masking is knowledge of intervention assignment is kept away from those directly involved in the trial. And people used to, and still do, use double blind, single blind. Please do not do that. Tell who was blinded and who was not blinded. Because, in fact, there have been studies where they asked clinician investigators who publish in clinical trials, on clinical trials, what do you understand by double blind? Oh, I was blinded and this was blinded, but what about the outcome assessment? What about this? What about the people treating the person? They don't understand those things. So blinding means everybody, whoever did the assignment, the participants, the staff, the clinicians, the study investigators, seeing the patients, the study statistician, sometimes the monitoring board, and many times the sponsor. They want to distance themselves. They don't want to know what's going on, because otherwise they said they interfered. And so some interventions are easy to blind. Some interventions are not easy to blind. Anything that involves a behavior like diet, you can't blind that. Anything that involves simple pills, maybe. But if they have side effects, then maybe not. Okay, so be careful what is blinded and what is not able to be blinded. So locations of masking is at the concealment, at the beginning, and then during follow-up. And in some studies, it may be impossible to blind during. So for example, if you're doing, several of you are interested in Ayurvedic treatment or in traditional Tibetan interventions, you can't blind that. But what could you do? When you cannot blind something, you have the person that is assessing the outcome be blinded, called blinded outcome assessment. There's somebody who is totally separate from the study, somebody who has never seen that patient before, who doesn't know what the patient was getting, who doesn't talk with the doctor, who doesn't talk with anybody else that had anything to do with that person, and they're assessing your outcome. So your outcome is very important. The outcome has to be assessed um, rigorously. Usually these things are done with, um, if you have a marker for it, biomarker, that's perfect. But if it's not a laboratory one, if it is self-reported outcome, that's a tough one. Because now the person knows what they're getting, and they know what you want to hear, or the outcome assessor, and they will say something that you want to hear. So it's really very difficult if it's self-assessment. You want to have some sort of objective way of assessing the outcome. And so there are instruments where you ask people to fill out things, but there's also tests. If you're, for example, want to look at fitness, there are measures that are quantitative, objective ways of measuring fitness. But if you want to measure satisfaction, there is no way. If you want to measure happiness, there is no way of doing it quantitatively that is not biased. Right? So be careful what you select as your outcome. But usually, if you cannot blind clinicians or patients, you can try to blind the outcome assessor and do that. That's what many studies do in situations where you cannot blind the intervention. Well, we talked about methods to conceal allocations, the mask during conduct. It's sometimes difficult. And side effects lead to unblinding. This was a study now fairly old where they had zinc or placebo for treatment of a common cold, but you had bad taste of the zinc, 79%, and here 30% of the placebo. So if you have bad taste in your mouth, you know you're getting zinc. So this is also a problem. So it's easy for, in this case, for the volunteers to know what they were getting. So what people tend to do at the end of the study, we do this thing where we ask the patient, what do you think you were getting? Were you getting active or control? And we ask the treating physician, oh, about subject X, what do you think he was getting? Active or control? What do you expect? You would expect, this is a study that was published in JAMA, that the truth was whether they were taken propranolol or taken placebo, and the patient was 80% of the patients guessed they were taking propranolol correctly, but only 57 did placebo. But notice that the physicians were slightly higher in this, not as high in that, but the clinic coordinators were the best in general. Why? Because they see the patient all the time. They know what they're doing. And so this tends to happen quite well. So people ask this, try to check that in masking was successful. And as I said, you should try to make sure that the outcome assessment, either if it's not a hard outcome, then you should try to get somebody that is external, not the staff or clinicians doing participant care or managing or administering the intervention. And this is a very important technique that many studies do in this situation. And we did a study where they were looking at multiple sclerosis, and we had two physicians that had contributed patients, and we kept them masked as to what they were getting. And so we said, can you give us your patients, whether you think they had certain multiple sclerosis, probable multiple sclerosis, possible multiple sclerosis, or doubtful multiple sclerosis. And this is what's called the agreement chart. And if these rectangles go along the diagonal, then there is no bias, and both raters are rating equally. But we see that rater B tends to give more certain and probable because they have more values over here than less values over here than rater A. And when it's his own patients, rater B's patients, he is very certain about most of them. And so he was basically unblinded. He could guess, even though we did not give names, we scratched off, and all they had was the symptoms. Oh, this must be Mrs. X. So she certainly has symptoms. So he was not really blinded. So be careful that your outcome event adjudicators were blinded or not, an outcome assessment. So summarizing this, is masking is a desirable design feature, makes the biases impossible and protects objectivity, may not always be possible, may not always be successful, and you should say who was blinded and who wasn't. So next, we want to talk a little bit about good clinical practices. And maybe you know about this, I'm sure most of you do. There is the International Council on Harmonization, ICH, that came out maybe about 20 years ago, 25 years ago, with guidelines for the conduct of clinical trials in a way that the European Union would accept trials that follow this. The United States FDA would follow trials that follow this, and the Japan Medical Council would follow them. And so the three groups got together and said, if you do a trial in any one of these places that follows these guidelines, we will trust those results. We don't have to do it again in our area. And they proposed that, and now many countries have adopted the same thing. India has the ICMR guidelines, and if you look at them, they are basically the same as the International Council of Harmonization. So these are guidelines that if you do your study following this, your results will be trusted by who? The government. Who is the people you want to convince that this is something that will change medical practice? So that's why it's important to follow these guidelines. So these good clinical practices are an international ethical scientific standard. They are not statutes or regulations. There is no police that's going to come after you. They don't have the force of law. They're just guidelines. You can follow them, you can ignore them. It's up to you. But if you comply, then the ethics boards will be happy. The public will be happy that the rights and safety of participants are protected, and the quality of your data is good. Aha, that's important, because if you do a bad study, you may have designed it perfectly, but you do a bad study, you have terrible quality data, you have a garbage. And you can do whatever analysis you want to do, you're going to be having beautiful, fancy models of garbage. You have a nice result, beautiful, fancy garbage results. Okay? So data integrity is very important. You need to have credible and accurate data. So what's the difference? Quality assurance and quality control. These are methods or procedures require this. You want to prevent the problems, you want to detect the problems, and you want to correct the problems. But as I said before, it's better to prevent them than to have to correct them, right? But you have to detect them. Don't create systems and never check, because if you don't check, I bet you will have problems. So quality assurance is all the planned and systematic actions that you establish that ensure that the trial is performed according with compliance to the regulatory requirements. And quality control are the actual techniques and activities that you do within your quality assurance system to verify this. Maybe slight difference in semantics, but when we do a study in a single clinical site, there's all sorts of jobs that happen, right? there's the principal investigator, a bunch of co-investigators. You have to have clinical support, you have to have statistical support, technical support, administrative support, and maybe laboratory support. We have all sorts of people that work here. Somebody gives you money to do it, unless you are very rich. Somebody, you ask for money. And then there's an external data and safety monitoring that oversees the study integrity. If you do it in a multi-center study, now each of the sites is now here. There are multiple sites here. And you may have centralized some of the activities. So now you have, instead of each site having its own statistical group, you have one statistical group. It's called a coordinating center. And maybe instead of having each site have its own lab, you may choose the central laboratory. Most of the times, this sometimes may not be possible if they're in different countries. For example, you may still have multiple labs, but you have to standardize them and calibrate them. It's usually better to have one that's calibrated, standardized, etc. And usually this is true, because this group then is checking the data of everybody and creates the same system for everybody, manages the data, does all of that. And the investigators from here are part of the directional group. So there is a steering committee that's comprised of the investigators from each one of these, and or some of the PIs and co-PIs. And if it's big, you may have some subcommittees to do different types of work on this. So this is sort of a general setup for multi-center trials. And you still have your data safety monitoring board. And again, you need a sponsor to give you money to do this. Now I make a distinction between multi-center, so these are all centers that are participating, from what I call a multi-site clinical trial. What's the difference? If you look back here, this stuff is the same. This is where the work takes place. This up here is the decision part. This is who decides what's in the protocol, who does all of the scientific part. When you do it as a multi-site one, it's the sponsor that does all of it. And the sponsor then contracts a bunch of people. You give me patients, follow my protocol. You do this for me, a CRO does this. You do this, a central laboratory does this. And they pay very well. And they give you, you give me patients, you give me patients, you give me patients, you follow my protocol. That's fine. But the decision, the protocol, the science is all here. These people are not really necessarily co-investigators. They are patient providers in some sense. And so they don't decide the policy or the publication. They may not even appear in publications whatsoever, because they're not invested in designing the protocol. The protocol is designed here. They just say, okay, I want to do this study. Let me find people that will do it for me, and we'll pay them to do it. So some people call this multi-center. I prefer the term multi-center for when there is science that is being conducted here, and they're part of the decision-making part. But that's just preference. People call these things, they don't make a distinction between. So the Statistical Coordinating Center in these studies is a group of people that have the ability to compare data from each collection site, make it easier to detect errors or problems than in a single center study, and are in charge of doing, this is what the CROs do, monitoring of sites. They have internal committees requiring quality assurance, external committees that provide oversight. There's statistical investigations that aggregate data. They're basically there to check data integrity and to make sure that data are collected properly, they're processed properly, they're summarized properly, they're analyzed properly, and they're presented properly. And so this is a big enterprise, the Statistical Coordinating Center. CMC Vellore is one of those statistical coordinating centers for clinical trials done in India, for example. Now, what requires quality assurance? Everything, right? Otherwise, why are you collecting it? You're doing an important study. Why are you collecting something that's not important? Why are you wasting your time with that? So everything that you collect should be important things, so everything should be of high quality. But most people collect more things than they should. So at least you should make sure that your critical variables like eligibility and your endpoints are done properly. You don't want to include people that should not be included, and you want to get the endpoints because you're going to judge the efficacy or effectiveness of your intervention based on the endpoints. So that better be done properly. Also, compliance with protocols that are critical to the validity, the reliability of each step of transfer of data, changes and clarifications to the protocol, documentation of ethical considerations, consent process. This is the kind of stuff that ensures that you are doing the study ethically and the study has integrity. And so people will check on you. Most trials that are done that are small, that are not done with pharma, that are not done with drugs, may never get checked or audited. But most studies that are done with drugs, with pharma, with devices, big studies, ones that have the potential of changing clinical practice, will most likely get audited. And being audited is a stressful situation because when you're audited, you are not follow. They don't follow the judicial system of innocent until proven guilty. They assume that you have done something wrong until you prove that you have not done something wrong. So you're presumed guilty of doing something wrong. And you have to show that you have not done something wrong. You have not tampered with the data. You have not done this. You have not done this bad thing at all. So you have to have proper documentation. You have to have what's called audit trail. In the old days, we had paper forms. And paper forms had to be completed in ink, not in pencil that somebody could erase and tamper with. And if you had to change something, you don't put white out. You scratch it, you change it, you put your initial, your date, and you are responsible. They'll come after you and say, why did you change that? And if you don't have that piece of paper and the original said it was a number 4 and on the database it's a number 2, tell me, how did it change from 4 to 2? You better be able to defend that. Otherwise, you're in trouble with the auditors. And the auditors are who? ICMR, FDA, these people, the regulators. So that's why we've been very careful to do it. So best thing is to prevent problems. How do you prevent problems? You have a well-written protocol. You don't try to collect everything in the world. Do a simple trial. Not let your friend down the hall say, oh, you're going to do a trial. Can you collect this other variable? Because I want to do a little study on this too. Get an echocardiogram on these people. No, but why? No, please, get. You start collecting bazillion things that are not important to your study. Takes time, effort, energy, money, and then you have to make sure that you don't waste your time with that and not waste your time on the things you must do. So don't do unessential things. Don't do complicated forms. Use uncomplicated criteria. Pre-test your forms. You need to make sure that investigators are committed to following the protocol. So if you're going to do this among your colleagues in different institutions, somebody says, oh, yes, Tambu, I will do it. But you know he's not going to do it. Then why do you invite him to do it? They have to commit. Friendships is one thing. Another thing is colleagues doing quality work. Be careful with this. So, I mean, we had one situation where this was actually real. This was a study of left ventricular dysfunction, and there were 24 sites, and they were planning the final protocol, and they were funded already to do it, but they had to finalize the protocol. And there was one issue about something, I don't remember now, on measuring injection fraction, one way or another. And one of the investigators at one of the sites said, no, I don't want to do it this way. This is not the way we do it. We want to do it this way, blah, blah, blah. They kept arguing. And everybody tried to convince him. And he said, all right, fine. I'll just say yes, but I'm not going to do it anyway. And unfortunately for him, that was in the minutes of the steering committee meeting. And when those minutes went to the National Institute of Health, which was the sponsor, I am not going to follow the protocol, was there. They took away his money. He was not part of the study. They have the big stick. When we're working with colleagues, we don't have the big stick. The controller is the money, right? So they did it. So it does happen. You better train your staff and certify them and check it. We had ophthalmologists there were 10 ophthalmologists in this study that had to, this was a study of keratitis. When you have herpes simplex that goes systemically, goes to your cornea, creates inflammation, goes away like all herpes, comes and goes, but it leaves an area of opacity. Eventually people go blind. What was important was to measure the area of opacity. And that was the primary outcome. And so you have here, this, and then the areas, but the areas may be something like this. Or the area may be something like this. How do you measure the area? So the statisticians, being great mathematicians, came with, I can fit an ellipsoid here, an ellipsoid here, and I can, with the formulas of the ellipses, I can figure out the area of the ellipsis, and I can subtract, and I can get the area. Fantastic. Let's just do that. Let's train these people to do it. And what happened was these 10 principal investigators were the heads of ophthalmology of major clinics. I said, what? You, statistician, are going to train me? I've done this for 25 years before you were born, or whatever. How can you dare to train me to do it? What do we do? So statisticians were clever. They said, they wrote separately to each 10 of them. Oh, famous Dr. X, you're so honorable. Please help us out. We are trying to do this study. And we need slides of people with keratitis. Can you send us 50 slides of your vast experience? Oh, yes. And they send you the slides, 50 slides. They asked it for all 10 of them. So they had 500 slides. And now they said, oh, Dr. X, famous Dr. X, please help us. We're trying to figure out the area of opacity. And we're not very good at doing this. You, with all your great experience, fantastic Dr. X, please do it and help us out here. And they sent 100 slides to everybody. But they didn't tell them that among those 100 slides, there were about 20 that were duplicates in random order of this. And they sent the same 100 to all 10. What did they get back? Enough to show that the area of opacity that one famous Dr. A did not agree with famous Dr. B or famous Dr. C or famous Dr. D. They didn't agree among themselves. And worse yet, famous Dr. A did not agree with himself. Because this is how they do things. Right? They think they know what they're doing, but they don't. And if you want to be objective, this is your primary outcome, you better measure it properly without noise. Remember, noise variability is the worst enemy. If you want to show that your intervention has an effect, you better have small noise and large effect. If you have a small effect, you better have very small noise. And if you have noise, that's the enemy. So be careful of that. And noise is lack of quality. So we calibrate the equipment, we calibrate the people, We have central laboratories. Then we maintain the trails and documentation for a potential thing. The when here? 5:30? Okay. All right, yeah, a few more slides. Then you want to detect it. We want to do central monitoring of data. So usually the statistical coordinating center will monitor the data as it's coming in. So we create usually the data entry system. Now there's commercial ones that are very expensive. We customize them. The CMC here creates their own data entry system also, customizing some software for it, making it look like the forms that you have, so it's user-friendly, etc. And it has all sorts of built-in checks, logical checks, consistency checks, extreme values. We sometimes do site visits to check without asking you, without telling you we're coming. We show up and say, oh, here's what we have on these patients. Let's see your records. Are these the same? Are these different? Did you make up data? Did you do this? You do it in a nice way. You don't accuse people, but you check on people. And they know sometimes when you come in and you sometimes not when you come. But then statistically, we look at aggregate data by site and by staff. So one of the things people don't realize that if I want to make up data, falsify data, cheat, I am not a good random number generator. I tend to do it differently, right? So I'll give you an example. We had one person who was taking blood pressures in that study of cholestyramine, the $120 million study. And those blood pressure was not a primary outcome, but it was a secondary variable of importance. And we all know that in those days, now we have digital things, but before then, it was the old sphygmomanometer, and you had to pump it and listen and say when, you say 120 over 80, 125 over 90, just like that. Nobody says 180, 123 over 79. So people have a digit preference. So they wanted to be more precise. So they had this fancy random zero machine. It was difficult to use. And they had to take blood pressure 8 times during the visit. Twice when the person comes in, 30 minutes later, twice again, twice again, twice again. So there's one guy who said, oh, I'm very smart, but I'm also lazy. I'll just take the blood pressure once. I'll add 2, subtract 3, add 4, subtract this, do this, do that. I can make up the other 7. He thought he could do that. And what we looked at centrally, we looked at the aggregate data and the variability. Many people think statisticians worry about means. No, you guys worry about means. We worry about variability. The noise is what's important. And if you're making up data, you think you're randomly generating it, but the variability that you do is much smaller than what is real variability. So we looked at the variability of the blood pressures of different staff members. One person stands out. He's so low. Now, we don't know if it's cheating or something's going on, so we tell the investigator, go and watch what this person is doing. This person was doing exactly that, was making up 7 out of the 1. And so the variance is reduced because he's using one number to generate the others. And he thinks he's generating it randomly, but he's not. And so what happened? Well, the person was fired. His data was thrown away. Luckily, $120 million was not thrown away. If that had been the outcome, the study is gone. Because one person does this kind of stupid thing. And we're all human. We're all lazy. We all look for the shortcuts. But here, we have the moral and ethical responsibility to do the study well. And that's not just the investigator. It's everyone. You as the investigator, you better make sure that the staff that you have, you trust them, you make them feel that this is their study. It's not your study. It's a study of the patients and the participants. That's who you're responsible for. But everybody that works in a trial, from the coder to the outcome measurement to the blood pressure to everybody, has an important task. And you better honor and expect that. So we call it accountability and responsibility. Each person is accountable for it, is responsible for the entire study. But then you don't say my study, it's our study. That term is very important. Okay? You still have to correct things. So you detect it early, and you implement procedures, etc., to address the performance and correct things as early as possible, and redress misconduct or fraud if that's the situation. So in different areas, you have different types of quality assurance things. Laboratories will have internal and external quality control. They also review distributions and outliers and determinations. You train the personnel, certify them. You document, and you have a data management plan. And we talked about incentives. We have also incentives for staff and sticks for staff as well. And so we usually don't control the sticks very much. Who has the big stick, whoever is given the money, they have the big stick, or the regulatory agencies. But people that are working together, the statisticians, we have very tiny twigs, maybe, the little leaves. That's all we can do. So clinical monitoring versus data management is management of investigators and staff. Management of subjects is important, that they follow the protocol as well. Regulatory compliance is important, and then quality assurance is important. I think I've sort of touched on all of these, and I'm not going to mention them again. So quality assurance is in every process, from recording the information, you check and correct the editors, then it goes to enter the data, then you check there, and if there's errors, you correct them, you append the interim database, and you back up, and then you come here till you finally get the database ready for analysis. Now, one of the things that's very interesting in this is that, and we'll talk more about this when we talk about how we do the analysis of a study, is that as we're collecting data, we're analyzing data. So this is not, we don't have the luxury in a clinical trial to do what we do in other studies. In other studies, what do? We say, this is the design, let's go and get the data, and I get all the data, and then I analyze. Clinical trials, we don't. In clinical trials, it's a moving target. As soon as we start getting data, because we want to make sure that it's clean, we better start cleaning it. And how are we cleaning it? We're doing all of this analysis to see that the data are good and corrected, because we don't want errors to continue to build up. We may have to train people. So we have to do this monitoring. It entails that we are constantly looking at the data. Oh, who is looking at the data? Not the investigator, the statistical group. All of those data managers, all the statisticians are looking at this, and maybe submitting it to an external board to see that things are okay. But we are monitoring it constantly, which means that our data systems are collecting data, and we need to get a little bit at a time. Well, this is continuing to get more and more data. We analyze this, and then we take another snapshot, and we analyze that, and we see what has changed, things that also changed, etc. So we have to have that system of freezing the database during the study, and then at the end, we'll freeze it at the end. But we have to freeze it to do the analysis, because otherwise, if I take the data today and look at it tonight and do some fixing, and then, oh, and I take another thing tomorrow, that have different data, because things get accumulated pretty quickly in these studies. So you have to have those systems in place. And this is a little bit more important, not only for the monitoring internally, but also for the monitoring externally. And when we talk about analysis, we're going to be talking about interim analysis that is done by others, not necessarily only of quality of the data. They're going to be looking at the safety of the participants and the study integrity. That is possibly the most important aspect of any clinical trial, is that report that goes to the data safety monitoring board. It's not any other report that you do. That one, I guarantee you, you better have it correct. If you're a statistician that is preparing the report, you better have that every number is correct in that. And it is important because the decision of maybe stopping the trial or letting it go may be dependent on something that you've done, or you've done it all. And if you've done it wrong, they may come up with the wrong recommendation. And so it's of utmost importance to have these reports done well. And we'll talk, I think we have a session on data safety monitoring board, so this will be, we'll talk about that. So I'll stop here now.